Now we will present EXPOSIGNALZ project. This project investigates the effects of chemical pollutants on neurodegenerative diseases, particularly Alzheimer's disease. The coordinator is Veronique Perrier here. Veronique, she has led research on gene therapy, diagnostics, and the impact of environmental pollutants on the brain. And she received the Thermo Fisher Biotherapy Prize and the, help me with the pronunciation, please, Triocholet Prize. Thank you very much. She currently leads a research group in the Montpellier Institute of Neurosciences on the impact of environmental pollutants on the brain across the lifespan, from neurodevelopment to aging, with a focus on neurodegenerative disorders. So the floor is yours.
So thank you, Christina, and thank you also to all the people who organized this kickoff meeting. So EXPOSIGNALZ project involves 9 partners and 14 teams in 5 different European countries for a budget of 9.3 million euros. We're very lucky to develop this project. The title is Dissecting Brain Pollutant Exposure and its Contribution to Neurodegeneration and Alzheimer's Disease, from Signatures to Prevention Strategy to Protect and Maintain a Healthy Brain. So our scientific challenge is Alzheimer's disease. Why? Because it's the 7th leading cause of death in the world. And in 2019, 57 million of people will be affected by dementia worldwide. And the projection by 2050 raised to 152 million cases. Among them, 60 to 70% will be Alzheimer's disease patients. Also, there is some hope right now because there is some disease-modifying treatment that was, disease-modifying treatment that was approved by the FDA in 2023 in USA and recently by the European Medicine Agency in 2024. Only patients in the mild cognitive stages, which means at the early stage of the disease, will be eligible to the treatment. Theoretically, it's 5.4 million people that can benefit from this treatment, but in the real life, only 10 to 30% of the MCI patients could have access to this treatment. So in our project, we want to focus on the development of prevention strategies, try to identify new risk factors, and prevent or delay disease onset. And according to the Dementia Prevention, Intervention and Care report of the Lancet Commission, there are right now 14 modifiable risk factors that have been identified and can prevent or delay 45% of dementia cases, excluding the genetic risk factor, which means that we have 55% of risk factor that remains to be identified. And the air pollution has been identified as a risk factor for Alzheimer cases. And the question is, how about other pollutants and what can be the role of pesticide in Alzheimer's disease pathology? The chemical exposome, can we identify a chemical exposome specific to Alzheimer's disease? And can we identify new risk factors? So the global aim of EXPOSIGNALZ project is to uncover how specific chemical pollutants affect brain aging and contribute to Alzheimer's disease and cognitive decline. For this, we have several objectives. The first one is to identify environmental pollutants likely to have neurotoxic effect and pro-amyloidogenic properties, potentially contributing to a neurodegenerative trajectory related to Alzheimer's disease. We have focused on a selection of 67 pollutants that we will screen in various in vitro and in cellular models. Among these pollutants, we have persistent organic pollutants, e-waste, mostly heavy metals, substances that are already monitored in water, pharmaceutical residue, pesticide, and some candidate that we are focusing on. It's the anilinopyrimidine fungicide. For the second objectives, we will characterize pollutant signature associated with brain aging, cognitive decline, and Alzheimer's disease in the human population. For this objective, we will benefit from 4 human cohorts, and so 3 cohorts in France and 1 in Sweden. And we will have 2 mass spectrometry platforms, one in Luxembourg to screen the chemical exposome in hair, and another at IRSET in Rennes. Globally, we will plan to screen the chemical exposome in 9,900 samples. The third objective is to understand the mechanism of action of this chemical signature identified in the 2 previous objectives in various Alzheimer's disease models. And for this, we will have cellular and preclinical Alzheimer's disease model. The cellular model are inducible pluripotent stem cells that we have already derived from Alzheimer's disease patients. And we can generate 2D and 3D models from these cell lines. And we have also 2 preclinical models where we can study the impact of this chemical signature on behaviors and Alzheimer's disease marker. The fourth objective is to understand how chemical signature identified impact early brain development as a susceptibility factor for neurodegenerative diseases later in life. For this, we will use mainly normal cells and wild-type animals models. We will use brain organoids as to study the impact on early brain development, and also maternal model of contamination to see the impact on behaviors of the animal. And we will focus also on exposure on all lifelong to see the impact on the age. So regarding the key expected results, we plan to do a ranking of environmental pollutants based on amyloidogenic properties, neuronal network disruption, neurotoxic effect, impact on cell viability, and blood-brain barrier integrity. We want also to do the assessment of environmental chemicals in human biological matrices, including blood, urine, and hair. We want also to do the identification of distinct chemical signatures associated with cognitive decline, subclinical Alzheimer's disease, or incident Alzheimer's disease. The results from this chemical signature will be investigated to understand the mechanism of action of this pollutant signature in Alzheimer's disease model. We will report the effect of this chemical signature on neurodevelopment and brain aging, challenging the developmental origin of health and disease concept. And we will also identify environmental chemicals as new modifiable risk factor for dementia incidence. Regarding the expected political and societal impact, we want, of course, to do the dissemination of our knowledge to scientific communities, specialized public, general public, and governance, and also the dissemination of knowledge to policymakers and regulators. We want to do some proposition of prevention strategy for healthy brain development and healthy brain aging, and proposition of guidelines based on relevant and robust data. And for this, we will have the help of one agency, Beta Technology, to help us for this dissemination and communication strategy. We will try also to identify the sources, high exposure group and vulnerable groups. We want to set up prevention strategy to delay the entrance of patients into the more severe stages of the pathology to reduce the cost. We want to reduce or delay the incidence of Alzheimer's disease and thus reduce the economic and social burden on societies. And finally, associate the earlier diagnosis of Alzheimer's disease with a preventative intervention strategy to, through our enhanced understanding of pollutants and their effects. So here is the logo of EXPOSIGNALZ, and if you want to have more details on the project, go to the website, and you will have further information on the partner and our project globally. Thank you for your attention.